Hello there, and welcome back to another episode of The Price of Pain. I'm Josh Crow, and just had a freeze here in Gainesville the other day. As I was on my way into work, I saw icicles hanging from some of the horticulture labs. Definitely a unique time. Put that together with Omicron, but nonetheless, Season 2 of Price of Pain rolls on. I've got an interesting guest today who is not just a brilliant researcher, she's also a nurse and a dear friend of mine, Dr. Keesha Roach. She's at the University of Tennessee Health Science Center, and she works out of the Department of Health Promotion and Disease Prevention. She's done a lot of great work already on racial disparities in pain, and she's had a large focus on sickle cell. But what struck me most in talking to her, aside from how impactful her current research is, and how over my head the genomics component of it is the fact that she's unique. It seems as if through her time throughout her undergrad, getting her degree in psychology and minoring in biology, and using that degree, working at the NIH and the Pasteur Institute in France, she's been all over the place and had so many experiences. But instead of choosing her career, it seems as if her career chose her. My only complaint about this episode is that we can't have these chats more often. So sit back and relax, and I hope you enjoy this episode with Dr. Keesha Roach.
Welcome to The Price of Pain, brought to you by the Pain Research and Intervention Center of Excellence at the University of Florida. Let's join host Dr. Joshua Crow in conversations with scientists, healthcare providers, and industry professionals as we delve into the highly subjective experience of pain and the ongoing effort to reveal its influence on our everyday lives.
So I've been getting just acclimated to my new environment, figuring out what the resources are and what the resources are not. I had to get my grants transferred over. I had to get, data use agreements, get IRB approval, like all of that time-consuming stuff.
The behind-the-scenes stuff.
Yes.
When you say the data usage, that's to use existing data up at Tennessee, or to continue to use data from down here?
Both. So some of the data that I'm using for my research is from University of Illinois, and some of it is from down here. So the majority of the data is from University of Illinois. So I did my data use agreement with them.
And what kind of data is that from Illinois that you're working with?
So when I did my pre-doc at University of Illinois.
Oh, yeah, I knew you had a connection there. Okay.
And I was working on a study. Had a diversity supplement on a study. And we did all of our data collection up there. I was still there, even though my primary mentor had actually come down here to Florida. I did all my data collection there, or all of the data collection there for all of her stuff and what I would eventually go on to use in my dissertation. And of course, now that I have this K01 grant, some of the questions that I've developed came from looking at that original data set.
Really?
So
that's pretty full circle.
Oh yeah.
I like it. Okay.
Oh, yeah.
Okay.
And so from that, I'm looking at a few other things within the K, and I'm adding like new, collecting data on new participants. Okay. And adding that to it.
And so for people who are looking at me scratch my nose, for people who are listening along, a K award is part of a publicly funded grant through the NIH. It's a structure, a specific structure. And there are multiple types of K award, but they're all, correct me if I'm wrong, intended to be for early stage investigators. Once you transition out of a training and into being an independent researcher, they're intended to kind of fund your research and your salary and whatnot in that phase, correct?
Right. So the particular K that I have, and I can probably safely say all the Ks, are training grants. And the type of K that you get depends on like the type of research that you're going to do. So if you're doing like clinical trials or clinical studies, it might be one type of K. And if you're doing basic science, it might be another type of K. And I'm looking at a diverse group. So that's related to the type of K that I applied for.
Okay. All right. And
Yeah.
so, and what specifically, so there's a training component, but then there's also a research component, right? You said with the
Oh, it
diverse
has,
group.
yeah.
So what are some of the training things? 'Cause this is something that we have all different kinds of people on the podcast from different points in their careers and different disciplines and research interests. I'm not sure that we've ever had a chance to discuss this. So what, just for you, if you don't mind sharing, what are the training aspects of your K? What do you want to improve in your toolkit as you move forward?
So one of the training part, all of it for me is training.
Mm-hmm.
So it's how to set up and start and run a study and do the data collection and do the statistical analysis. And I'm doing some epigenetics training. So learning how to use Python in different programs that'll allow me to at least, I don't plan on becoming a biostatistician per se,
Mm-hmm.
but when I'm looking at my data and looking at the output and thinking about writing another grant and what's going to be needed, I need to at least be able to have a conversation with the statistician.
Right.
And so it's important to me that I learn like at least some of the basics of what that is. So I'm learning that as a part of my training grant. And the beautiful part is that I get to have a mentoring team. And as I come up against, so just really for everything, as I come up against barriers
Mm-hmm.
that might occur, I can talk to them about it. I can talk to them as a group if it's going to take all of our brains,
Mm-hmm.
or I can talk to them individually because each of my mentors has their own specialty.
Right.
So one person does genetics and genomics, and that is where her doctorate and all of her training there, she's a full professor. And another one, her training is in pain specifically,
Mm-hmm.
and patients with cancer and patients with sickle cell disease. And then my third mentor, her training is also in pain, but it's pain and cognition. And so all of these for me, as they relate to sickle cell disease.
I think that's awesome. In that, first of all, you're very well rounded as it is. You know, with your degree in nursing, and didn't you have a minor in psychology or something like that from undergrad also? And
No, I had a degree in psychology.
you had a degree. Pardon me. Okay.
From the University of Maryland.
Look, as great as UMD is, and I got my degree in psychology from Arizona State. They're all great. Undergrad degrees in psychology will get you so far, right? But so, degree, a minor, there's a small difference, right? Anyway. But in all seriousness, you've got a well-rounded skill set as it is. So I think it's really cool to think about and to share that actually with our audience, that just the nature of research and researchers, and that you're always looking to piece more skills into your overall skill set. And one of the best ways to do that is to get a platform that you can kind of stand on there to reach out to experts who've spent their whole career on that. So, you don't have to have an infinite expertise in genomics. If you get to the point where you have a firm foundation, then you can communicate with somebody who's devoted their whole career in that and then collaborate.
Exactly.
Right? I think that's awesome. That's awesome.
And my path was, I call it the long and windy road because
you and me both.
I started off in psychology.
Mm-hmm.
With a minor in biology. So
Okay.
that was my minor.
Okay.
And I worked at the National Institutes of Health during my last 2 years in undergrad.
Mm-hmm.
And I found that I really enjoyed learning about genetics. And this was like back in the day because I started off when they were doing restriction fragment length polymorphisms.
What is that? Because most people, me included, have no idea what that means.
That's when you take smaller pieces of the DNA in order to like get information about what is going. So now we look at like the full genome, but in this one they were using specific enzymes to cut the genome and then look at these smaller fragments and then put them together and see.
Just to see what the smaller fragments did on their own.
Right.
Okay.
Right.
What are we don't do that so much anymore, and please educate me along with everybody else. Throughout this conversation, I'm oftentimes going to act as a proxy for our audience because I'm sure that if I have a question about it, then somebody out there is also. Sometimes that means I have to ask questions I already know the answers to. Mm-hmm. Sometimes I have genuine questions. So this is a genuine question. you say that we've gone from looking at a piece to now looking at the entire genome. What are some of the benefits of looking at the whole genome? Why is that an advancement? Why don't we get what we need to know from looking at the individual pieces?
That's a really good question. But what we can see looking at the entire genome is we get more answers. So we get more answers, a bigger picture of what's happening, what we think that we're seeing. And also we have the opportunity to, as other things come up, if we, for example, don't have all of the information for a specific gene that we want to see, we can now take from all this other data, impute, and get information about the gene that we didn't necessarily sequence.
I see. By finding out what's not going on,
what's around it. Yeah.
Okay.
So that's definitely, I think, one of the pluses of using the whole genome. So.
Yeah, it's one of those things where, and it's funny because when you think to put your expertise in layperson's terms, it's funny. I've come upon this before and gotten feedback from our audience as well. Stuff that you've done that's so commonplace now that seems like, oh, well, they already know that. A lot of times people don't. You're just getting so used to using it all the time that like you have to scale way back to the point where it's like, oh, I get it. I see what you're saying. So any of these questions, I'm not trying to stump you by asking a hard question that I think sounds simple, but that really doesn't have an easy answer. So I'm not trying to put you in a spot.
No,
but there's so much of this stuff you've already forgotten, regardless of where you are in this path down genomics. You've already forgotten far more than I'll ever know about this topic. So you have to bear with my ignorance.
No, that's fine. I mean, right. And then it makes me think about God. What is this?
Why don't we do that anymore?
How do we describe the fork versus the spork?
Right? Oh, okay. See, that's a great analogy. Okay, good. So, all right. So let's go back then just a little bit, because not everybody gets a chance to work for the NIH. Were you actually in Bethesda or?
I was in Building 10
wow.
when that was the building.
Wow. That's so cool.
Yes.
That's so cool.
So I was with the first year, because I did 2 years. The first year I was with the National Institute of Alcohol Abuse and Alcoholism, and under Dr. David Goldman. He's still there, still amazing, still in contact with him as needed. And then the second year I was in the National Mental Health.
Okay. Institute. Okay. With what'd you say the first one was? Alcohol and substance abuse?
Alcohol abuse and alcoholism. NIAAA.
Okay. And what specifically were you looking at with gene segments and genomics and what? Well, I guess it was genetics.
Well, for him, so I was definitely just coming in as a research assistant, cold off the street, literally. I wanted to work there so bad. I would look, I got I put in an application, I didn't hear anything. And I put in another application, didn't hear anything. And I went and got the catalog and looked at all the people who were doing work that looked interesting to me. And I just went to their office. So the first person that I went to, we had a conversation, and he was like, told the person to get the paperwork going.
Oh, that's awesome. You wandered into the right office.
I wandered into the right office.
That's such a cool story. So, but it doesn't stop there. So you spent time working there, but then you also got a degree in nursing, but the degree in nursing is not your PhD.
No, the degree in nursing is my PhD. So
oh, it is.
When I left after I finished my undergraduate degree, I did not stay in the DC, Maryland, Virginia area. Okay. So I left the NIH. And I took a position at University of Chicago in the Howard Hughes Institute there, working in another lab where they were doing molecular genetics research. But this time it was in diabetes. And once again, I really enjoyed my time there. I enjoyed working, and late hours. My family knew to call the lab before they called my house. And it wasn't because anybody asked me to stay late. That's where I wanted to be.
Oh, that
great. Was like my sweet zone, my zen zone.
Do you know how many people would kill for that? to be able to make money doing exactly what they want to do, or to build a career and a degree exactly what they want to do.
Yeah.
A lot of times it starts off that way, but I don't know if it's ever as intense as what you're talking about, where
It
you're
was
just,
it was good. But for me, it wasn't like I was doing my own research. I was just doing whatever experiments that the team needed me to do.
Mhm.
Um,
And gaining
and then
experience the
running
whole
the
time.
lab and getting experience the whole time. And then if summer people came in for the summer, the students, to kind of manage them and make sure they learn all the things in it they needed to learn to, excuse me, to get to do the work that they were asking them to do, basically.
Mhm.
And then I followed the work that I was doing. It was a collaboration with a French group. Followed the work that I was doing to France, because when they finished the U.S. portion of it, the French team wanted to continue their leg of it. And since I had already been working with those DNA samples and things, they found a way to get me a carte de séjour and get permission for me to come and work in the lab at the Pasteur Institute, even though I didn't have a PhD.
So for lack of a better term, that's the French connection. Because we've talked before about your time in France and how you loved living there. I didn't realize how that came to pass. I thought it was a familial thing, like you have family over there. No, you're just in the field crushing it, and they're like, "Yep, you know what? You're so good at this. We should send you over there and do a little bit more."
Yep. So they kept the research assistant / coordinator, and so once again, it wasn't my own work.
Mm-hmm.
I only had a bachelor's,
Mm-hmm.
but I enjoyed doing, and I was learning the whole time.
At what point did you decide that you wanted a PhD then?
I knew I wanted a PhD from when I was in high school.
Oh, really? You're one of those people.
I was like, yeah, that's where I'm going to end. But I couldn't see how to make it happen.
Mm-hmm.
And when I would talk to people who I thought were so amazingly smart and bright, they would always say, well, it's really hard. Are you sure that's really what you want to do? And I would, oh, really? It's like, well, so I didn't ask the right questions.
So did any of that take hold then? Did that actually discourage you from getting the PhD?
It discouraged me from getting a PhD.
That's a shame.
But a part of me always wanted it anyways. I would always circle back and think about that.
Mm-hmm.
And in fact, so I'm fast forwarding.
That's all right.
So there was a student, and one thing that I tend to do is I love to help people who are trying to make the next steps in their lives.
Mm-hmm.
So a lot of, high school kids come to me, or young kids, or can you help me with my application for college, or can we talk about X, Y, and Z? So I helped a kid do his application for college, and he got accepted, and then I helped him negotiate what his financial aid package should look like.
This is while you were just still with your bachelor's degree doing science. You weren't a counselor.
Yeah.
You're doing okay.
Yeah. And then I decided at that point, I was like, okay, Keesha, just face it. You haven't done the PhD you haven't done it. So just forget it. Move to the next level in your life and just forget it. And I literally got physically ill when I made the decision not to get a PhD. Really? So that next day I filled out my application.
All right. So you know what? That is awesome. And I'm going to tell you, I'm going to share a little bit of a story of my own. But I love that because you made a decision, and the reaction of that decision is what led you to one of the most important actual decisions you could have made, right?
Yeah.
I do this on a small scale all the time. Sometimes I get indecisive. Now, this is not me minimizing or belittling your choice. I'm just saying, when you said that, I was like, oh, wow, so I'm on to something here. On a much smaller scale, I'm sitting at home like, man, I don't know if it's getting kind of late. Should I cook dinner or should I just order pizza? I flip a coin. But this is the thing. I don't necessarily go with the answer. I go with how the answer makes me feel, just like you did.
That's right.
If I say, okay, I'm gonna go ahead and get off my butt and cook something. B, I'm gonna order a pizza, or heads or tails rather. So I flip a coin, and if it's tails, I go, nah, I probably should have cooked something. Then I go cook something.
Right.
You know what I'm saying?
Right.
So
Exactly.
that's sometimes, no matter how brilliant you are, case in point, you have to follow your gut.
You have to follow your gut.
Okay.
You have to follow your gut. And one thing that I did in between there, before I even applied to this PhD program, I had come back to the U.S. and worked a little bit more as a research assistant, and now they gave me I had a title of like scientist 2 or something.
Okay.
At a biotech company out in Torrey Pines California.
Mm-hmm.
And I felt the inkling of, I've been doing this for so long. I haven't moved forward with it. I haven't gotten a master's or a PhD. But my burning question is, how is all this work that I'm doing tied to, like, how does that look on the human side?
Mm-hmm.
So that's what made me get a nursing degree.
Okay.
And I knew that I wanted to get a PhD, but I felt like I could better inform any research question that I had if I had an understanding of like how this directly impacts the human side.
So you're saying then you wanted, and that's maybe that's where I got confused about your PhD not being the nursing degree, because I know you've said before the reason that you went into nursing was specifically for that. You wanted to see what was happening at the patient level. You wanted to see the end point of all of this work, and so that's why you got into nursing.
Right.
But you went with the whole shebang anyway. You went PhD nursing, so you could do both the clinical component and the research component at the same time.
Right.
Okay.
So I went into nursing and I stayed in the field. I planned on staying in the field for like 2 or 3 years and then going back for my PhD, but I ended up staying in the field for 15 and then going for my my PhD in the College of Nursing. And and in the meantime, I was thinking, well, do I want to do this in nursing or should I do it in something else? And really, I looked into more than one type of program. But what I learned is that nurse scientists had come so far in what they do, and they did everything I had, and even outside of nurse scientists, just nursing period. I had friends who were nurses, but then they went to University of Chicago and got their JD, so they were RN JDs.
Oh,
and
wow.
Nurse friends who had MBAs and run businesses, and so I got the idea that I wasn't going to be limited if I chose nursing. But I knew that, the funding might be there. I knew that I might be able to finish if I stayed focused.
Right. See, that makes me want a nursing degree. I feel, and I all the joking earlier about the psychology degree, a psychology degree alone doesn't do career-wise a whole lot for you. You always need something in addition to that. But the benefit of that degree to my development, at least, and how much it's informed so much, even once I got into applied physiology and kinesiology in graduate school, has been so beneficial for that. But it sounds like the nursing degree is another one of those types of degrees that just has a way of kind of bolstering anything else that you do.
Oh yeah.
Okay.
Oh yeah, for sure. So for me, it serves to drive all the questions. In fact, because of nursing, I do sickle cell research. Like, no one in my family has sickle cell disease. I didn't know anybody personally. At least I didn't think I did. I later learned that I definitely did.
Yeah.
But it took one patient. I was going to do diabetes research. That's what I knew, and that's where I was comfortable. It took one patient in the emergency department one night suffering from vaso-occlusive crisis.
What's that? Break that down for us.
That's when your red blood cells are, you're in a low oxygen state or a stress state, and your red blood cells change shape, basically, and they go from being round to being a sickled shape or a C shape, which is where sickle, the name came from.
I see.
And then these cells go on to clog the veins, and you can have a vaso-occlusive crisis anywhere. You can have it in your hip, you can have it in your brain. So you have like babies that have strokes at a little age, at a young age, 6 months, 9 months, because of sickle cell. You can blindness, priapism. You have problems with your spleen, splenic sequestration. You have vaso-occlusive crisis in your lungs. They call it acute chest. Any place where the blood flows, you can have a crisis there.
So in sickle cell anemia, the red blood cells are not always in that sickle shape. They get depleted of oxygen, and that's what changes them to that shape? Yes.
So they're not always in that shape, and depending on the severity of your sickle cell disease, kind of determines how if it sickles or how long it sickles. Or if it sickles or how long it takes before the hemoglobin sickles. But one of the things about sickle cell disease where you don't see it in really young people is because when your fetal hemoglobin doesn't sickle.
Mm.
And so after you've gone through the part of your life where you've depleted your fetal hemoglobin,
Mm-hmm.
that's when you start to see, crisis, vaso-occlusive crisis, if those individuals are going to be prone to that. Because not everybody who has sickle cell gets vaso-occlusive crisis.
Okay.
It's just so many variations, and that's where all this research comes in.
So can you not diagnose sickle cell until that change occurs? Then once all the fetal hemoglobin is, so like you no longer have that in your system? Is that what you're saying?
No, you can. So we haven't always, but we now have prenatal. We have prenatal testing. So
Okay.
when you're born, you get tested to see for many things, but one of the tests is to see if you have sickle cell disease. So I think it started like in the 70s when they did the prenatal testing. So from that, you have your results.
I'm curious. I want to scroll back just a little bit here. You said you planned only on practicing nursing for a couple years before going on to your PhD, but it turned into 15. Was it this that kept you there, or was it something else?
No, it was something else.
Okay.
I enjoyed like working in the acute care setting.
Okay.
So I was in the cardiovascular ICU for a while, and the medical ICU for a while, and the neuro ICU for a while, and so for each one, it comes with a different skill set.
Mm-hmm.
You're learning new things, and not only are you learning new things, you have to implement them quickly. It's fast paced. You only get 2 patients or 1 patient per shift because the patients are so acutely ill in the ICU settings.
Okay. So that gathering more knowledge and moving on to the next thing and learning a different system and a different type of care, that's what kept you there for a little while longer. So then what you mentioned, the one patient with sickle cell that really said, "Oh wow, this is intriguing to me." What was the thought? Was it the question mark of why, or was it how they were treated or not able to be treated? What was it that really turned the rudder for you? Not only, I mean, at that point, when you came upon that patient, were you already working on your PhD, or was it during that clinical phase?
Oh no, I was a relatively new nurse.
Okay.
At the time.
Okay. So what really turned the rudder then toward the interest in sickle cell anemia?
Because he was sitting there suffering in the emergency department, and nobody seemed to really care. And these were people that I was working with, and people that were training me as a new grad. I was learning from them, like, what do we do? How do we treat these individuals? And their main response was that he was faking.
Interesting. So when that patient came in, what symptoms were they exhibiting? What did they report to you that, I mean, did they say, "Hey, I have sickle cell anemia," or did they say, "Oh, my chest hurts," or what was it that they thought that they could test versus what they thought the patient was faking?
So when I came on to my shift, the patient was already in the emergency department. He was already in a room.
Okay.
So I got report at change of shift, and I was working the night shift that night, and he had gotten some pain medications, but he was saying that it wasn't enough. That's not what he normally gets, like the amount that normally works for him. And he said what the amount that normally works for him is. And they said, "See, he's even telling us what to give him." And they use that as an example of him being drug seeking instead of an example of him having lived with the disease long enough to have an understanding of what typically worked for him, because it wasn't his first vaso-occlusive crisis. And I remember, like, he was watching television, and they were like, "See, he forgot that he was complaining about pain. He's sitting there watching television." And I was like, "Okay, look, know that I'm a relatively new grad, but one of the things that we learned about was distraction. Like, how are you saying this? And so it was confusing to me. I was in orientation too. It was
not really in the position where you could speak out a whole lot, right?
I mean, I could, and I did, and I ended up going to a different institution.
Because of that?
Yes.
Oh, wow. Okay.
Yes.
So this is a big moment for you. Let me ask this really quick also. Young guy, older guy?
He was probably in his late teens or early 20s.
Oh, man. Okay. So, and this
and
is...
he ended up throwing his tray at me.
Oh, really?
He was so frustrated, and all I could say is, I see you. I see you. I see you.
Yeah. Wow, that's powerful. And so you got into that degree path in that environment specifically to learn about what happens at the patient end. And you got an answer that was pretty poignant, it sounds like, particularly with sickle cell.
Mm-hmm.
But also, we've had other guests on, be it Dr. Star Booker, who's mentioned this, Dr. Emily Bartley. We've talked about, Dr. Chris McCurdy, who was, just recently on for the second time, about differentiating between people who really know their condition and what treatment they've had in the past because they have to live with this every day, and drug-seeking behavior, which is a very real thing, and how difficult it must be to delineate between the two and recognize the signs of each. And so what were some of the things that you saw in this young man that really let you know, hey, we're not getting it right here?
I think that what the main thing that stood out for me is that from my training in nursing school, is that we're not the judge and the jury of somebody's pain.
Mm-hmm.
That pain is subjective, and pain is what they say it is. And we have to treat the patients with the information that they provide us with.
Mm-hmm.
And with their history of our patients, we can see like previous admissions and things like that. So I don't know if that was his case. If that was his home hospital, I don't think it was.
Mm-hmm.
But, if he had been admitted in the past, certainly there was going to be some kind of record.
Sure. Now, when somebody comes into the ED that is complaining of pain, are there tests, or maybe I should roll back to that point. Were there tests that you could do to get an idea if they were in a severe vaso-occlusive state that would, taking all subjectivity of pain out of it, that would say, okay, this is definitely going on right here. If he's reporting pain, this would make sense that he's in pain because we can see that he has sickle cell, and right now he's vaso-occlusive. Are there assessments like that you could have done at the time, or that you did do?
So when a patient comes in and he has a diagnosis of sickle cell, especially if they've been to that particular hospital before, that the report of having sickle cell is real.
Mm-hmm.
But no, you have to just constantly assess the patient and go by what you see.
Mm-hmm.
And sometimes their response to pain medications, if that's what they're getting, if it's enough or it's not enough. And in some cases, you can speak to their physician if they have a physician. Even if the physician's at another institution, they might call in and speak to whoever's the attending and
Mm-hmm.
have that conversation. But the other thing is there are certain algorithms that are in place for people who come in with pain. If a guy comes in with chest pain and shortness of breath, then there's a cocktail of what he's going to get or she is going to get to address that pain, because it could be a heart attack.
Right.
And they'll know, like, for sure after they do not for sure, but they'll have a really good, estimation that this is what's happening.
Right.
If after they do like an EKG, for example.
With sickle cell, can you get an indication of what's going on by looking at pulse oximetry, like looking at oxygen saturation?
No, but there are other tests that they can look at. For example, they can look at like the platelet count.
Okay.
They can look at the white blood cell count.
So it's more on a blood analysis level, not something that you can really see from the outside.
Yeah.
So, well, okay.
And
So that stuff doesn't do you very much good in that moment. You really have to listen to what the patient is saying as far as their pain.
yeah, and the labs will come back, and the labs are not, the end all, but it at least helps to guide.
Mm-hmm.
And even sometimes if, with the labs, they may or may not listen
Mm-hmm.
to the patient. I'm just trying to think now how much things have changed, because I've been out of that setting for a long time. And in some places, in some cases, things have moved forward.
Mm-hmm.
In some places, not so much. I mean, sometimes you have physicians in the emergency department treating patients, and they've never seen sickle cell before. So, they don't really understand it or know what it is. The place where I am now, they have, between the different hospitals, or actually a few specific hospitals, they will treat the patients who have sickle cell disease, and they will keep the records. They'll have like an algorithm of what happens, and it's patient specific.
Mm-hmm.
And if that patient's not from that hospital, they have like grand rounds between the sickle cell providers from the different hospitals and say, hey, we saw your patient.
Right.
That's very unusual. And it's great for the patients, but that's not how most places work.
I think it was you that we had a conversation a while back, long before this episode. And I think it was you, correct me if I'm wrong, but you had said that, I want to say it was one of your research participants, told you that they were asked either by a nurse or a physician, oh, when did you contract sickle cell?
Yeah.
But you don't contract sickle cell.
No,
That's not how that works. You're
born
born with
with
it. So that just kind of highlights the fact that this is, why do you suppose it's
That's
not
like, hey,
Yeah.
when did you contract blue eyes?
Yeah. So why do you suppose that is then? Why is there that huge gap in knowledge in a profession that really should have their finger on that pulse, no pun intended?
It could, I don't know. I speculate, and I don't know if it's because, I don't know if I'm jaded or just I've seen things and look at them from a realist perspective. I don't know if it's because, of the population that it impacts,
Mm-hmm.
that the level of caring just isn't where it needs to be.
Mm-hmm.
And more times than not, I think that that's what it is. And I hope to be wrong.
So then you say the population, who predominantly has sickle cell then? What population?
So sickle cell disease predominantly impacts people of African ancestry.
Okay.
So, it impacts people of African ancestry for sure.
Huh.
And predominantly in our country, African Americans.
Mm-hmm.
And then to a lesser degree, people of Latino descent.
Okay. All right. I was going to ask that actually, because that's one of my blind spots when it comes to sickle cell. Do Caucasians, do white people can they, I don't want to say get sickle cell, can they be born with sickle cell?
So in like the Mediterranean region,
Mm-hmm.
You do have white people who get different types of sickle cell, like beta thalassemias and the different levels of thalassemia, which when you have two parents with different types of maybe thalassemia, you might end up with one of those forms of sickle cell that's deleterious. That gives you a lot of sickle cell crisis. That's very painful.
So there's more than one type of sickle cell then?
So there's sickle cell, but there are different types of genotypes within the sickle cell. So whether you have, I wish I could explain it better. I wish I had been more prepared for this question.
Not at all.
But like,
this is a casual
you have
conversation.
SS, and then you have SC, and you have SE, and you beta thalassemia plus, and then you have beta thalassemia negative, minus, and did I say E, D? So there are about 6 or 7 different types of sickle cell within your genotype.
Okay. And so they can combine based on your two parents and you get different flavors of sickle cell that could deform the red blood cells that you have based on certain conditions.
Right. And then other things impact, whether or not your blood cells get sickled and your how your veins respond and whether or not you get crisis. For example, stress is one of those things. So you asked about, we were talking earlier about my degree in psychology. Well, my K award, the title of that grant is Biopsychosocial Factors of Sickle Cell Disease Pain. Because I'm looking at, behavioral and things like stress and resilience and what are your sleep patterns and then more bio, like what are the inflammatory markers that might be floating around, what concentration, and then genomics, what genes are related to pain and which genotype are you expressing? Is that more related to more stress and just all these different things that
we have and that's fascinating, and it seems like it's could very easily be a lifetime worth of work because we have all of these factors to talk about the biopsychosocial model. We've discussed this on the podcast before, where there are different influences, biological influence, psychological influences, social influences that can affect how an individual, not necessarily even a type of person, but how an individual subjectively experiences pain. And all of those factors, and they're not always constant, right? There are some, they can be, some of them are state dependent, meaning how you are right now. Some of them are more trait dependent, right? So that's how you always are. But then when you take something like sickle cell that seems very complicated unto itself and overlay that over just the pain model that everybody experiences, no matter who you are, that's a lot of different combinations. It seems like a moving target, I should say, when it comes to diagnosis and to treatment and to looking at how you can really help these patients.
It definitely is. One case in point is at one of the institutions where I worked, there was a sickle cell center where the individuals would be able to come in. They would be able to bypass the emergency department and see someone if they were having problems. And we had a person who would come in pretty regularly. We knew who he was, and it wasn't all, sometimes it was dehydration, sometimes it wasn't always a need for pain medications per se as to why he was coming into, but it was related to his sickle cell. A lot of times it was pain, but not always. And one particular time, the physician put the patient into the room and, I'll be right back. Then he comes out into past the waiting area, and he's like, "Hey, didn't I just put you in the room? How'd you get back out here so fast?" Turns out the guy had an identical twin, monozygotic twins.
Oh, wow.
Of which the twin never had a crisis.
Really?
Yeah. So, and we were like, that would be a great case study.
Yeah.
And I don't have more information.
Because he was an identical twin, he had sickle cell, but that never manifested into the pain and the claudication and everything that comes along with the cells actually sickling.
Right.
Wow, that's fascinating.
Yeah, it is. It's very fascinating.
But also perplexing. Because how do you, if that's the case.
To your point, it's very wide and deep.
Well, you chose the right career path, my friend. There's a lot of work. You know what that says to me? Job security. So what?
Pulling my hair out.
Right? See, I can't get into situations like that. I don't have a whole lot of wiggle room when it comes to hair pulling. So tell me a little bit about what you're working on specifically right now then. You had a fantastic job talk that I had the pleasure of sitting in on when you were here. Before you left University of Florida, you were doing, as scholars do, developing the presentation that you would give that talked about your background and your interests and your funding with your K and where you were headed. And it was so fascinating to me. And I would love, even if it's just a little bit, now I'm not asking for a presentation by any means, but kind of clue me in with where was it a COMT gene that you were looking at? Man, you got to help me out because this is so far in the deep end of what I do. Like, I'm a meathead scientist, so you need to help me out with this, but peel back the curtain just a little bit
Okay.
on what you're doing right now, what your current interest is in, because I know I just totally butchered that. But with sickle cell and sickle cell pain.
So we did, the group that I was working with, so the group who definitely mentored me and took me under their wing as I was coming up, they had some data on a CompT DRD3. Yes.
All right. So I was right.
So that is a paper that we wrote together.
Mm-hmm.
I was first author. I did the, background, look at the work and wrote the paper, and and they of course scrutinized it, and you go back and forth until it's ready. So so we did that. But one of the main things when I first applied to my PhD program, I knew that I wanted to do genomics.
Mm-hmm.
So it was important for me to find a group that was using, and I knew that I wanted to study pain. So it was important for me to find a group that was doing that. And I was lucky. At the University of Illinois, I did find that.
Mm-hmm.
And so just because I was getting started again, I had been away from the bench for a long time.
Mm-hmm.
I mean, I was in the hospital for 15 years.
Mm-hmm.
So, there were they had just determined a couple of pain-related genes that had like high frequency within the African American population.
Okay.
So I picked a couple of them and just started learning about it and doing some research on it in terms of looking at the pain and how this related to pain and hospital utilization, average pain intensity in relation to this particular gene. And so we found that, with this gene, you didn't necessarily have a lot of, I can't remember the CPI. I can't remember the term for that.
Right.
But you
layman's terms are totally cool for this group. I'm going to act on your behalf. Yeah, it's okay to keep it at the pain by numbers level with us.
You did have acute pain, and you had a higher level of utilization that was significant.
Okay.
So I went ahead and looked a little further at that and looked at the same gene in older and younger adults. So what is it? Is there something protective about this gene, or, what is it with? And then we also related it to stress. So we didn't use, it's because it was retrospective in a sense. We didn't specifically use a stress scale, but we used other proxies that pointed to stress.
Like what? Like give an example of how you could do that, how you could tease out stress.
We went to, in the comment section, when you can write down anything you want. So some of the individuals wrote that they were having stress, and that the stress, that they related the level of stress that they were under to the reason why they were having pain.
Let me tell you right now, just hearing that is making me stressed out, because when it comes to working data in a study, it's really nice when everything falls into a scale, a numeric scale, or a yes and no, or, check the box. When you have to go back and read comments, I don't know how many participants you're talking about and what kind of sample size, but that's stressing me out just hearing about it. That stuff alone is not easy. But please, by all means, now that you've got me all worked up, continue.
Well, actually, it's not that bad. You go and you look at the comments, and then you rate for the people who talked about stress or no stress, you just rate it as a 0 or a 1.
Okay.
If they get a 1, if they talked about stress being related to the pain, and then 0 otherwise. And then, of course, for that kind of data, that kind of research, you get someone else to also look at the data without knowing what you've done and see what's the consensus. And then you come together and you have discussion about the ones where there wasn't a match, and then you get your final result. So,
besides, you get the undergrads to do the 1s and 0s for the comments, right?
Well, I was a pre-doc,
so that was you.
So that was
all right. Okay. Well, you've spent your time in the trenches. Then it's all easy from here on out.
I definitely did. So we looked at stress, and we saw some indicators of this particular gene, this particular genotype, and stress. And then I looked at another sample of individuals against this gene, but this time I broke it up into younger and older adults. And I did that because I was just curious as to whether there was something protective about this ABPR1A gene or this particular genotype within this gene.
Now, are all of these participants that you're looking at, are they all African Americans?
They're all African Americans.
Okay.
They're all African Americans with sickle cell disease.
Okay.
So
What, if I may ask just really quickly, and you may not have this off the top of your head because it was a while ago, but is there a specific age range? Were you looking at older adults, adults with sickle cell, everybody from teens upward? What kind of, and both sexes equally, or?
Yeah, so they were age and sex matched.
Okay.
Or age and sex matched. That was another study. But anyways, they were they were all adults. The younger adults were age 18 to 39.
Okay.
And the older adults within sickle cell disease because of the way that because of the life expectancy, basically.
So how does that affect?
It's 42 and up. Well, patients who have sickle cell disease they're not expected to have a long life. But we have people who are living longer, people who, for various reasons, we've had patients who lived into their 70s, and even here in Florida, a colleague told me of a patient that they had in their 80s who had sickle cell.
But that's a really big indication of how sickle cell can impact lifespan, because look, I'll count myself fortunate if I live into my 70s, but also there's a big part of me that's expecting to do that, right? That 70s is not old anymore.
Right.
However, if you're talking about, well, some of them live into their 70s, then that really is a huge impact on longevity.
Yeah, because it can just as easily be, well, some of them live into their 50s, because we lose a lot in their 30s and 40s.
Now, what about, and I know you have to please forgive me for taking this tangent, but this is also something that I think a lot of people don't know about sickle cell. I certainly didn't. As far as affecting lifespan, when it comes to, I hate to use the term, normal adult. So let's just say a healthy adult. Pick any race. When it comes to a healthy adult, you have a lifespan where, if you're fortunate and healthy, your younger years, you're pretty resilient. Your adulthood, you get your aches and pains and stuff. But each stage of life is marked by some pretty specific characteristics, right? When the average person, if there's such a thing, gets older, okay, so you may have more musculoskeletal pain, some more joint stiffness and soreness. You may not have the endurance that you normally have. You may not have the balance that you once had. But these are all characteristics of later stage life. If you're looking at someone with sickle cell, on the other hand, what does their late stage in life look like characteristic wise, whether it's in their 30s or whether it's in their 60s or 70s or 80s? Is it akin to?
It varies a lot with each individual. It's very individual. But you have a lot of you have individuals who start to have regular aging factors, and then you have others who've had like vaso-occlusive crisis and pain issues, or maybe they have had some deleterious like stroke or something like this happen that they're having to live with, even though maybe they're not having as many crisis. So there are things that compound.
So I was going to say, that's another thing, that as you age, your cardiovascular health typically declines. If you're 75 years old, you say, oh man, your heart looks great for a 75-year-old. But if you compare it to a 20-year-old, it's a 75-year-old's heart, right? So since we're talking about red blood cells, as a person's vascular system ages and those changes, those natural changes occur, compound that with any lifestyle, behavioral, dietary changes, I can see how that could very easily compound the effects of these vaso-occlusive events. Yeah. And so you have somebody who's dealing with this more frequently, more pain involved. What?
It just depends on the individuals. Just in this year alone, 2021, one, so not this year, last year. Sure. Of the individuals that I know of who passed away from sickle cell disease, maybe there are about 9 or 10.
Oh, wow. Okay.
And they were all in their early 30s to mid 40s.
Oh, man.
And that's just me not keeping track.
Sure, that's just anecdotally, just
Yeah.
your people you've come into contact through work or friends or.
Right.
Wow.
So, for example, for the pain gene that I was looking at, that's turned out to be related to stress and pain in sickle cell disease. We're wondering if there's something protective, because you can look at this genotype and break it up between younger and older adults, and the older adults are more likely to have the protective genotype than the younger adults.
And explain to me again why you think it's protective. Is it because they exhibit less pain? Is it because they have fewer vaso-occlusive incidents? Yeah. What is the gene doing?
It's because they tend to have fewer vaso-occlusive incidents. They tend to have less pain, and they report less stress than the individuals who have the different genotype within this particular gene. And so, then we went and we looked and we're like, where is this located? Well, it turns out the gene is in the promoter region. And we're like, okay, well, what the promoter region is.
Yeah, it's just good. You catching on.
Is the region of the DNA sequence that is where the proteins are, like, if it's how do I put this?
You tell me.
So what it does is it regulates the amount of protein that's produced.
Okay.
So it's either going to be produced to a higher extent or to a lower extent. You don't know what part of it really turns it on or off, or whether it produces more or less.
Okay.
Those are other studies that you look at.
Right.
But the interesting part that it's in that region.
Okay.
So one of the things that we're thinking about looking at, or have started looking at, is like, based on your level of stress and pain, what does your protein profile look like? With the timing of when we collected this data, this patient was having high pain or stress. So that's just something that we're looking at.
Is it possible, and I know these questions are, this is a layman question, because oftentimes when I ask them, it's not known. But I'm curious, to try to, at least I try to entice our guests to speculate a little bit. But is it possible that this is part of the genome that could be activated by stress, or is it that it's protective of the negative effects of stress on somebody with sickle cell? Is it possible that it could be either one?
It could be either one. Okay. It's possible.
So how do you get to the bottom of that?
So when we're looking at like the different protein profiles and seeing what like the amount produced, and then relate that back to the patient's current state when you collected the data, and then also looking at other proteins around it, like what else is being impacted and being produced or not produced in these situations.
Now, you know my background a little bit. I think people that have listened along to the podcast, usually the focus is not on me, so maybe not so much. But most, I think, understand that I'm a physiologist to some extent, a kinesiologist to another. I have to ask then, exercise is often a panacea, right? It alleviates many symptoms and improves many conditions. But with exercise, part of exercise is physiological stress, and it's the adaptation to that physiological stress. So the stress that you're talking about with individuals who exhibit this particular piece of the genome, when you say stress, do you mean emotional stress, physical stress? Is it both? I mean, how do people with sickle cell respond to exercise?
Well, I suppose so to answer the one question,
yeah, that's a lot. I just threw a lot at you.
I'm looking more like at emotional stress, because with emotional stress you can have visceral reactions.
Right.
So that's one thing, but also with physical stress, I haven't so much looked at physical stress. That's something coming down the line when I'm looking at, like, to answer some of my questions, I might use ecological momentary assessment.
You might need a collaborator, is what you're saying?
Yeah. Mm-hmm.
Okay. I know somebody. Did I give you my card yet?
I know somebody too. Yep. So for those, with respect to exercise and sickle cell disease, a lot of groups are looking at that, like how are the patients able to tolerate it?
Mm-hmm.
And once again, it's not just a one size fits all. Unfortunately, it's never a one size. It's not that easy.
Right.
They're not gonna let us off that easily.
That seems to be a reoccurring theme that comes up no matter what discipline the guests we have on the show, their expertise is in, is that there's such a need for precision medicine. Mm-hmm. that this treatment is going to be specific to you. Now, that may fall within a region of people like you, and it can be tweaked a little bit. But at the end of the day, just because you look this way and talk this way and come from this place and are this old, that doesn't mean that this pill is going to work for you, that this treatment's going to work for you. We've got to make sure that it's specifically for you. It seems like sickle cell is another one of those domains.
Oh, definitely.
More job security. I wanted to ask you something, though. You are working, and this is something that we'll talk about off air as well, working toward making a connection between the podcast and a colleague and friend of yours. And one of the things that we talked about when we were setting this episode up is you wanted to have her on because of what she does here at UF, if I remember correctly, but also a little bit from her perspective, from the patient experience, again, for people who have and are suffering from, I should say, sickle cell. What are some of the things, just, in closing, because we have only a little bit of time left, what are some of the biggest issues that people living with sickle cell have to deal with that somehow get lost in the shuffle when it comes to the patient care side of things that you alluded to earlier? You mentioned pain, but are there some other issues that people out there are really, screaming at the top of their lungs, like, hey, why don't we have fill in the blank? Why don't you get fill in the blank? Or can you share some of those things?
Well, one of the main things are the wait times in the emergency department. If I come into the emergency department and I'm telling you that I have so much pain and I have a diagnosis of sickle cell disease, why am I still, and you're telling me that you care? Because some hospitals have liaisons that kind of try to, either, I don't know, just hold a job or actually try to bridge and help the patients. But, if a patient with another condition comes in with excruciating pain, they're less likely to have to wait as long as a patient who has sickle cell disease. And so I don't know why that is, because it's everywhere. But
we do know that according to the data, and this is something that's very widely reported, if somebody who looks like you reports an 8 out of 10 according to pain, and somebody who looks like me reports an 8 out of 10 of experienced pain, I'm more likely to be taken seriously that I really am at an 8 out of 10.
Oh, yeah, absolutely.
So it is, and you had alluded to this earlier, but just to make it perfectly clear, when, just, I guess to be blatant with it, when someone who is black says, I'm in pain, that's not taken as seriously when compared to someone who is white.
Oh, yeah.
The problem is, in this case, pain is a symptom of a disease.
Right. Pain in sickle cell disease is a symptom of a bigger problem.
Right.
So just because you came in last week with vaso-occlusive crisis, maybe you had vaso-occlusive crisis, and maybe you had vaso-occlusive crisis and it was impacting your shoulder joints. But maybe this week it's impacting your hip joints. Are you leaning towards having avascular necrosis? This week, another week, you're, reporting pain in your abdomen. Is it splenic sequestration? That's a medical emergency. And if you're just sitting in the emergency room waiting for somebody to see you, you could die in the emergency room. Right. And it has happened.
So that's, I mean, and just to be clear, that's akin. If somebody comes into the emergency department, they're in the emergency room and they are pale and they're not breathing, and, let's say the person that brought them in says, oh, they grasped their chest. If you take one of those signs and symptoms away, the whole they're not breathing thing, well, now all of a sudden it could be something other than a heart attack, right? Mm-hmm. So, and this is my non-medical analogy I'm making here. My point is in many of these cases with a sickle cell patient, pain is a very important sign or symptom.
Yes, absolutely.
And so you cannot ignore that, because then you could misdiagnose and maybe incorrectly triage them in threaten their life even. Mm-hmm. Sounds like a big deal to me.
It's a very big deal. It's a very big deal.
So what are there groups out there? Are there advocacy groups for sickle cell that are working to try to promote awareness, not only amongst physicians, but amongst the public as well?
Yes, it kind of depends on where you are. So, for example, here, Florida, the state of Florida is the second most highest prevalence of sickle cell disease in the U.S. But where I am now, in the tri-state area, Memphis or Tennessee, Mississippi, Arkansas. Okay.
Tri-state, quad state.
Tri-state, Tennessee, Mississippi, Arkansas. It's very dense. So as far as being, the most populous, well, it's not. But in density, you can throw a pen and hit somebody, piece of rice. You don't have to look very far.
Right.
So, it just.
Well, that's a great place to be conducting research then, and to be making contact and advocating for those groups, obviously, because you have the opportunity to help a lot of people in that area.
Right. So my point to that is the critical mass, because so many people are impacted by it. Mm-hmm. You have, their Sickle Cell Association, for example, where people participate in the walk, and it's not just a small group of people, it's a big walk, and the newscasters get involved, and different personalities, the public personalities get involved. Mm-hmm. Whereas here, where in this area where we are, where it's not as densely populated, the community support, just from me being a layperson with respect to that, the community support does not seem to be where it needs to be. But there are fighters out there. There are people out there with and without sickle cell disease, even here in the Gainesville area, who are trying to impact, make a difference to help the patients who have sickle cell, even if it means that they have to come to the emergency department when somebody is in the ED, and they might give them a call and say, nobody is believing me. Can you come and help me? And there'll be somebody who can come. But that's a situation where you have to know, you have to have a relationship, so to speak. Yeah. So it just varies a lot. And there are groups of people who want to make a difference. But I think that at some point you have to have a critical mass. And there are national organizations as well. So all we can do is keep our fingers crossed and keep working towards it. Yeah. And some areas have the momentum now. Mm-hmm. The area where I am, they have the momentum because there's so many people impacted.
Well, thank you for your work in this field. And I love the fact that you took the time throughout the progression of your career to stay on the clinical side and to be that person who is a patient's first contact. And, I get that it was eye-opening for you, but in listening to you, that wasn't an accident. That was something you specifically set out to do. And that takes a very special person to then take that and segue that into a very, thus far, successful research career. So, thank you for that, and also thank you for taking some time at very last minute. You just found out that you were coming down to Gainesville, on Friday?
I did, Friday night.
Well, thank you for reaching out, because you're a guest that I've been trying to get on the show for quite some time now, and I hope that this is just the first of many.
I hope so as well. I'm looking forward to doing this again.
Thank you for joining this episode of The Price of Pain. Opinions expressed on this podcast are solely those of the host and guests, and not representative of the University of Florida or parent institutions of our guests, unless specifically stated. You can find more information about Price on the World Wide Web at price.ctsi.ufl.edu. And keep up with our researchers on social media by searching Facebook for UF Price, by following @UF_Pain on Twitter, and Price of Pain Podcast, all one word, on Instagram.